Hello, everybody. Welcome to Ms. McGuire online video lecture, and we continue covering chapter Beyond Mendel's Law. Now, we already covered the first part of this PowerPoint, and we cover these three outcomes. And today we will talk about mitochondrial trait and inheritance of linked traits. Right, so let's go ahead and begin. But before we continue, I just want to remind you that when Mendel studied his garden pea plants, he looked at characteristics or traits that were inherited by those plants from parents. And those traits were located on a different chromosomes. They had two distinct forms. For example, I'll give you example. Allow us to understand better. So Mendel looked at pea plant, for example, the one with yellow seeds and one with green seeds. So he had two parents, one parent that always produced green seeds and one parent always produced yellow seeds. He crossed them and he looked and like, okay, my F1 generation of offspring from the first cross have green seeds. That means green is dominant over yellow. So that pretty much, it's very simple representation because we have several genes that determine our traits. We don't have one single gene that determines how tall we are. We have many genes that work together to give you your specific traits, right? Or we have genes that don't have complete dominance. It can be incomplete dominance. It can be codominance. We have genes that interfere the expression of our genes. So the inheritance is way more complicated than the ones that Mendel presented us. But it doesn't mean that Mendel's laws are wrong. They actually, using Mendel's law, we can explain all type of inheritance, even beyond his laws, right? Because Mendel didn't know anything about genes, about chromosomes. But now, by getting more knowledge about biochemistry of our DNA and RNA and proteins and different genes, we're still using our current knowledge and Mendel experiments, and everything works together. One does not contradict another, right? Now, this chapter specifically talk about phenomena that seems to be exceptions of Mendel's law, but are actually not. And we already talked about gene expression before, like how lethal alleles affect the ratio, how codominance, incomplete dominance, epistasis affects the phenotype. And it seems like it doesn't work the way Mendel predicted us. But no, this is just beyond this law, but everything still truth about what Mendel told us, right? So we talk about gene expression. And today we will talk about other genes that we have not in our nucleus. So most of our DNA is located in the nucleus of our cells. And of course, when we reproduce, what is that cell that gonna be the embryo and the fetus? That's gonna be an egg, right, or ovum, and a sperm. So when sperm fertilize an egg, we create zygote, and this zygote then divide and grow into entire organism. Now, inside this zygote, we have DNA, and in that DNA, we have 46 chromosomes. So most of the DNA, I'm sorry, what did I say? So we have zygote. Inside the zygote, we have nucleus, right? Inside that nucleus, we have DNA. And we have 46 chromosomes inside the nucleus. And that's the most of our genotypes. But we also have organelles called mitochondria. Mitochondria provide cellular energy, and mitochondria has its own DNA called mtDNA. This DNA makes mini chromosomes with only 37 genes. The inheritance pattern and mutation rates for mitochondrial genes differ from those for genes in the nucleus. Mitochondrial genes are maternally inherited because when we have ovum, right? So ovum is the female gamete. Ovum has genetic material, right? So, this is the egg or ovum. It has 23 chromosomes only, remember? And it has all these other organelles. So that's Golgi apparatus, Raffler, Smoothera, and it has those mitochondria. Sperm has only pretty much genetic material, only 23 chromosomes. So this mitochondrial DNA found in the mother egg, so that's why it's maternally inherited. Mitochondrial genes, let's see. Yeah, okay. Mitochondrial genes are transmitted from mother to all her offspring. So you can see if mother has some mutation in the mitochondrial DNA, it will be passed to every single child. So this, that. So it doesn't matter if it's a boy, so that's a boy, that's a girl, boy, girl, boy. So mom gonna pass it to all her children, right? So now this is a dad, all right? So he get it from his mom, but because he's a father, he's not gonna give it to any of his kids, right? So that's a boy, the son of this mother, he inherited this mutation, but sperm doesn't carry mitochondria. So you see, he is not passing it to any of his children. But the daughter of this mother gonna pass it to all her kids again. So that's a different pattern of inheritance, right? Very interesting. A cell typically has thousands of mitochondria, and each has numerous copies of its mini chromosome. Mitochondrial genes encode proteins involved in protein synthesis and energy production. Several diseases result from mutation in mtDNA. Example, mitochondrial myopathies, weak and flaccid muscles, or Leber optical atrophy, impaired vision. Theoretically, a woman with mitochondrial disease can avoid transmitting it to her children if her mitochondria is replaced with healthy mitochondria from a donor. So that can be done, but theoretically at this point of time. Okay, so that's also affect the pattern of inheritance, right? When we're talking about mitochondrial DNA, it seems like it has nothing to do with Mendel's law. Why? Because Mendel didn't know about mitochondria. He didn't know about mitochondrial DNA. And because mitochondrial DNA only passed from mother to her children and never from dad, from father to his children, the pattern will be different from a simple Mendelian inheritance. Another phenomena that seems like doesn't follow Mendel's law are linked genes. Linked genes are genes that are close on the same chromosome, and then they are linked. They're not assorted independently in meiosis and usually inherited together with the chromosome as package into a gamete. Do not produce typical Mendelian ratio. So here's the example. So remember when we talk about independent assortment, I give you example. If I inherited eye color from my mom, does it necessarily mean that I'm going to inherit hair color from my mom too? Or I can inherit my mom eyes and my father hair. And we know, yes, I can inherit my mom eyes, my dad hair, my mom, I don't know, nose shape, and my dad's ear shapes. So that means you inherit characteristic independently, But this is only true if they are not located on the same chromosome. But what if my eye color and my hair color are located on the same chromosome? Like, for example, that's my dad chromosome that I get from my dad, and it has eye and hair color that are brown, for example, and mom has blue eyes and blonde hair, and they're located together. Then look, through the meiosis, right, this is one chromosome, two sister chromatids. So here the father chromosome, and this is the mother chromosome. So in this situation, these genes, they are not independently inherited. So if genes are located on the same chromosome and they're located close to one another, then they inherited together, right? So eye color and hair color will be inherited together. That's interesting, right? So that's what called linked genes. And so you can see how parents different if genes are on the same chromosome, on a different chromosome. So this is the genes on a different chromosome. So eye color is on this chromosome and hair color, for example, on a different chromosome, right? So that's parental, this is maternal, paternal and maternal, sorry, paternal, maternal from dad, from mom. And when they are independently inherited, you can get all different combinations over here. So you will get this 9:3 to 3:3 phenotypic ratio. But when they are on the same chromosome, then dominant P always goes with dominant L and recessive P always goes with recessive L in this example. And then it's a different phenotypic ratio. So that means that Mendel's law of independent assortment really works only for chromosomes or for genes that located on different chromosomes, or at least they located far away from each other, and they can be there will be a crossing over between them, right? So this is a recombinant. What does mean recombinant chromosomes? Chromosomes recombine during crossing over in prophase 1 of meiosis. A new combination of alleles are created. If we have original configuration of genes on a chromosome, we call those parental chromosomes. If we have new combination of alleles, we call it recombinant chromosomes. So here's the example. So here's the chromosome. So where we are. So let's imagine that we inside testes, right? So there is a guy, and we inside the testes of this guy, and there is a cell inside the testes that's gonna be a sperm. So here's the sperm. And now the cell need to divide through meiosis to make sperm. Remember, we have meiosis 1, we have meiosis 2. Now this guy who we're talking about, he has his genes and his chromosome from his parents, right? So he gets this from his dad and this from his mom. Then he replicated the chromosome. So this is one chromosome. It has two sister chromatids. And now look, has some genes, for example, dominant P, dominant L, recessive P, dominant P, dominant L on one chromosome, and recessive P, recessive L, recessive P, recessive L on another chromosome. So when this chromosome cross over, see, they exchange the fragment, and all of a sudden, we should divide it, sister chromatids should split, and those are linked genes, they should stay on the same chromosome. So you see how P and L, those are linked genes because they are on the same chromosome. If you think it's about eye color and hair color, they kind of should go together, right? But because of this crossing over, we create recombinant chromosomes that now, look what it has. It has the little p, recessive P and dominant L all of a sudden, or recessive L and dominant P, the one that parents did not have. So we still have parental chromosome, dominant with dominant, and recessive is recessive, and we have 2 recombinant chromosomes. Now, even if you're not going into detail of all this process, you can see that pattern of inheritance became more and more complicated when you start taking into consideration those linked genes. All right, so you have now 4 different type of sperm because of this recombination. So even if genes are linked, so what we said before, we said if those genes are located on the same chromosome, they should be inherited together, right? Dominant with dominant, recessive with recessive. So this should be our sperm, shown right here. So this should be sperm. 2 dominant, 2 dominant, 2 recessive, 2 recessive. However, because of crossing over, we have this type of sperm. We have here 2 dominant, 2 recessive, and we have 2 recombinant chromosomes, dominant with the recessive. And that's all need to, and geneticists, they need to look at where exactly those genes are located and predict how they can cross over. What is the possibilities that they will cross over, right? So that prediction of the human traits became more complicated than the simple inheritance described by Mendel. So allele configuration is important. If we have 2 dominant or 2 recessive alleles on each chromosome, we call it cis chromosome. All right, so for example, right here, we have a chromosome with 2 dominant and chromosome with 2 recessive. So that's going to be our cis chromosomes. When we have 1 dominant and 1 recessive on the chromosome, we call it trans chromosome. And when we look at frequency of recombination, remember when chromosome cross over and we have recombination of genes. So when we look at frequency of recombination, the further those genes and alleles are apart. So look, this is allele and this is allele. They kind of, those are allele for gene A and B. So we have gene A and gene B. And then we have dominant A and recessive A, dominant B and recessive B. So genes A and B are far apart from each other, so crossing over more likely. So crossing over can happen here, can happen here, can happen here, or here, or here. We're still gonna separate. So if crossing over happen here, this part goes there, and this part goes to maternal chromosome. But when genes B and C, they close together, then they crossing over less likely, because you can, if crossing over must only happen in this spot. So the recessive C goes to blue chromosome and dominant C goes to pink chromosome, right? Here we can have crossing over in many places between A and B. So when we look at this frequency of recombination, we can see the correlation between crossover frequency and gene distance, and it is used to construct linkage maps. So this is how linkage maps work. So for example, we have 3 genes, right? We have gene X, Y, and Z, right? So, and we know that X and Y crossing over 10% of the time, X and Z crossing over 4% of the time, and Z and Y crossing over 6% of the time, right? The frequency of recombination shown over here. Then you're making a map. So you see where they're located on the chromosome, right? So here you have a chromosome. So now you have a chromosome, right? And you need to see, well, where this my X, Y, and Z are located. Is it they're located like here I have X, here maybe I have Z, and this is where I have Y. Are they located like this? Or maybe I have Z's here, Y is here and X is here. So relative to each other, what exactly the allocation is? Then we're looking at frequency, because X and Y recombinates, there is 10%. So that's a bigger, well, the highest percent of recombination. So that means they're further away from each other. And X with Z 4% and Z with the Y 6%. So we know exactly how those genes are allocated. So we will have X, right? We will have Y further away, and we will have Z that is more close to X than to Y, right? Because this is frequency recombination is 4, and this frequency of recombination is 6%. So we will have not even this type of recombination, because Z should be closer to X than to Y, not in the middle. And this is what this is how they form the linkage maps, right? So we need to know where chromosomes are located relative to each other, and by knowing the frequency of their recombination between the genes, we build the map, and this is how we can figure out where genes are located within a chromosome. I think that's the last, yeah, this is the last slide. And I just want to remind you that we're covering patterns of inheritance beyond Mendel's law. And we talked in this lecture about mitochondrial inheritance and linkage. I thank you for watching, and I hope it was helpful.